Hi everyone, so we'll be doing a few questions from the experiments that we have practiced, we've learned about in the last few days, right? So also with that, I'll tell you a few tips that you can use, common sense tips basically, nothing to do with bio, which you can use to answer your questions in NEET as well as any other competitive exam as well, because the kind of syllabus that you are given, it is very obvious that you might forget 10% of it by the time you reach the exam center. But you can still use your expertise that you will gain over 2 years of learning the subject, which you will use with your common sense to answer correctly. Right? So let's start with the first question. If 50 heavy and 15 DNA are replicated twice, right? So now let's see, you have N15, N15, you replicate it once and you replicate it twice, right? And if you have 50, then in the first you will have into 2, that is 100, and in the last you will have into 2, that's 200. So you will have 200 in the second generation, 100 equali in the first generation. Now from what we learned from Meselson Stahl, what should happen? In the first generation you should have N15, N14, right? Because they will both distribute themselves, and then N14 will incorporate. And in the next one, you will have N15, N14, because they will, so this will form an N15, N14. Right? And this will form an N14, N14. Correct? So, in the first one, you had 2 copies of this. In this you will have what? You will have how many copies? You will have 2 copies of this, and N14 get 2 copies. So basically you will have 100 N15 N14, and you will have 100 N14 N14, right? That should be your calculation. So let's see which matches our answer. A is 100 N15 N14, 100 N14 N14. So that's your correct answer. Huh? Now, let's look at the other options. In the second option, you have 100 N15 N15, जो आपको पता है possibly नहीं है ना? क्योंकि conservative तो होता नहीं है DNA replication, और conservative नहीं है तो original N15 N15 तो conservative हो नहीं सकता. तो ये option तो वैसे ही गलत है, मुझे पता ही लग गया. इसमें भी N15 है, तो ये भी गलत है, और ये तो ना किया हुआ. This was not right. Correct? So now, once you will eliminate these two, you will have 2 other options left. है ना? आपको पता है ये है, तो आप ये टिक कर दोगे. अगर ये नहीं होता तो none of the above होते, है ना? So what this is called is, it's called the process of elimination. आप अपने options को eliminate करते हो, ताकि वो 50-50 वाला concept that happens in KBC, right? So you are given 4 options, उसमें से 2 हटा दो, तो आपकी probability बढ़ जाती है of getting a correct answer. From 1 out of 4 it becomes half, right? Now, next question. A DNA molecule in E. coli is labeled with N15, right? It is allowed to replicate in N14. After 1 generation, the daughter molecules will be, मैं इसको लेने इसी से करेंगे. तो एक generation के बाद, this should be the case. What should be the case? You should have 2 intermediates, no heavy, no light. Now let's look at the options. Be similar in density but different from parent. Yes, they will be different from parent. Both of them will be similar in density. So yes, this can be right. है ना? Different density as well as that from parent. This part is correct. It will be different. But दोनों के densities तो same होंगे ना? 2 intermediates से मिलेंगे, तो ये तो गलत है. Has same density as that of parent? Obviously wrong, because conservative नहीं है. Different density but resemble parents. Again, can't resemble parents. So again, A is the correct option. Yeah. Now, if bacteriophage having S35 infects a bacteria growing on normal medium, the bacteriophage is produced after death of the bacteria will be. Okay. So now बैक्टीरियोफेज में S35 से इन्फेक्ट किया गया है बैक्टीरिया, राइट? सो योर वायरस हैज प्रोटीन ऑन द, I mean has radioactivity on the protein port, and it goes and infect the bacteria. The DNA will go and the protein port will stay out. Now when they will go and make new viral particles, तो यहाँ पे जो lysis होगी, तो जो नए बैक्टीरिया आएंगे, क्योंकि उनका DNA अनकोटेड था, उसकी प्रोटीन भी अनकोटेड होगी, है ना? So therefore, what will happen is that there will be have S35 in their nucleic acid? No. Will have S35 in their particles? No. Will not have S35. So C will be the correct answer. There will be no S35. Because obviously the protein coat was coated or had heavy isotopes in it, and therefore वो बैक्टीरिया को इन्फेक्ट करने के वक्त पे वो रेडियोएक्टिविटी पास ऑन ही नहीं होती. Okay. Moving on to the last one. By using protein from TMV RNA and from HRV. अच्छा, protein from TMV and RNA from HRV. Okay, TMV is tobacco mosaic virus. RNA is also another virus which causes leaf rot in plants. A chimeric virus was made. Chimeric मतलब दोनों को मिला के hybrid virus we can call it chimeric. It was injected into tobacco leaves. What did they find? So now we know that RNA can be inherited because of nucleic acid. TMV में protein cannot be. So TMV का कोई भी infection tobacco में नहीं जाना चाहिए, but HRV का चलेगा. So therefore we will find an HRV infection in the organism and not TMV infection. So all other options are there obviously wrong. Right? So on this chapter, usually the highest weightage is in Meselson Stahl's experiment. But however, छोटे questions from Griffith, from MacLeod, Avery, from Hershey are very, very possible. Another thing that you have to remember when you solve these questions for boards is that wherever possible, you should always give diagrams. So for Hershey, often a question comes where you have to explain why semi-conservative model was accepted. For that, if you look at my video, you will see that I have drawn. So I understand it might be time consuming, so don't draw the DNA bands, but at least draw the predicted DNA results and the centrifugation results. It doesn't take much time, but it helps you score a lot more, and you have to write less. In biology, when you are writing larger answers, you should write less and draw more, because drawing more gives your answer sheet a better clarity for the teacher to read, and therefore helps you score better. Okay. From the next video, we will start structure of DNA and DNA replication. I hope this much was clear. Thank you.